So proteins are these gigantic, very complicated molecules, these huge polypeptide chains. And so when we talk about proteins, it's going to be helpful that if we can consider them at different sort of magnifications, different levels of their structure. So the most zoomed in way to consider a protein is to actually look at the sequence of individual amino acids that are bonded together by the peptide bonds. And that's what we call the primary structure of a protein. So actually knowing the exact order of the 100 or so amino acids that make a protein chain, that's the primary structure. Okay, but oftentimes what really matters for the function of a protein is more zoomed out than that, and sort of how it behaves as one gigantic unit based on its overall shape. And so the amino acids, as they have linked up by these peptide bonds, they still have those R groups that stick out from the chain. And those R groups can interact with each other based on different types of IMFs or even forming covalent bonds with one another. And that causes the protein chain to fold and curl and do all these interesting interactions with itself that gives it a very complex three-dimensional shape. And that complex three-dimensional shape is what we refer to as the overall tertiary structure of a protein. Okay, so it's the overall three-dimensional shape or conformation of a polypeptide chain. This shape includes or may include what we call secondary structures, which I've skipped over and I'll get to next, because I think tertiary structure is a bit easier to understand. The protein's function, especially globular proteins, which we'll talk about in a subsequent video, things like enzymes, is incredibly dependent on its overall tertiary structure, its shape. The tertiary structure is maintained through a number of different types of attractions, notably hydrogen bonding, London forces, so two of the IMFs. Notice that dipole-dipole forces is not on the list because anything that is polar in the R groups is actually capable of forming hydrogen bonds, not just dipole-dipole forces. So it tends not to be on the IB mark schemes. So try to remember that the interactions for tertiary structures does not include dipole-dipole structures. It can include ionic bonds, ionic interactions, because as we've talked about, amino acids can pick up charges. They can be, their R groups, if they have amines, might pick up hydrogens and become positively charged. If they have carboxyl groups, they might lose hydrogens and become negatively charged. So different R groups can have different charges, and those opposite charges can attract ionically. It's possible for a certain type of bond to form called a disulfide bridge, which is a specific type of covalent bond. This is going to happen especially between the amino acid cysteine with other cysteines. And cysteine has an SH on it as its R group, or part of its R group, which is quite unique. It actually has sulfur. And if two cysteines get together, they can actually, the two sulfurs can bind, the hydrogens can leave, and it forms this bridge. It's called a disulfide bridge because it's formed of two sulfur atoms bonding together covalently. So it's quite a strong interaction. In terms of biochemistry, when we talk about covalent bonds, that's going to be a fairly permanent type of attraction that's quite difficult to break. It's also possible for different R groups to form peptide bonds with one another. A peptide bond, again, is an amide or a carboxyamide link, and it's going to form through condensation. So some of the R groups have carboxyl groups on them, and if you've got a carboxyl group and then you got another R group nearby that has an amine on it, so that'd be H to N, another H to begin with. And so if these two groups are near each other, they can condense, removing that water and forming the peptide bond between them. So these types of interactions would be the strongest types of interactions that can hold together different parts of a tertiary structure, but these forces are there as well. Okay? So secondary structures are, they're sort of in between the primary and the tertiary. They're specific three-dimensional shapes or structures that occur regularly within the overall structure of a protein. And they occur entirely due to hydrogen bonding that we would call intramolecular hydrogen bonding. So a little bit different than what we've talked about before, intermolecular hydrogen bonding, because the hydrogen bonding is taking place between parts of the same molecule. and therefore it's called intramolecular hydrogen bonding. The two shapes that you should know by name are the alpha helix and the beta pleated sheet. Okay, so an alpha helix is sort of a coil. Okay, so it's like a spiral. And so alpha helices results when there are a bunch of R groups near each other on a chain, about 3 or 4 amino acids apart, which can hydrogen bond with each other. So you can see these dotted red lines in the diagram represent different parts of the chain that can form hydrogen bonds with one another. It doesn't just have to be the R groups, but the particular sequence of amino acids has to have these, every 3 or 4 amino acids, there has to be something that can hydrogen bond with part of the chain nearby. And that causes the chain to sort of coil up on itself very tightly so that these hydrogen bonds can interact, and it gives this characteristic helix structure. And when we talk about the different types of proteins, fibrous proteins, which is one category of proteins, are composed largely of alpha helices. The beta pleated sheet, on the other hand, is a little bit different, and it's going to be formed when you have sections of the amino acid chain which can form a lot of hydrogen bonds with another section of the chain. So I'm going to draw sort of a line through here. So the stuff on this half of the line, of my red line, would be one section of a polypeptide chain. So these are actually the amino acids all covalently bonded together. And then some other portion of the protein chain is down here, and it just so happens that all of their R groups can line up and form hydrogen bonds, which keep these two sections of the protein holding together quite tightly. And so it ends up being sort of a flat section of the protein. That's why it's called a sheet. But it's also a bit pleated, and pleated means it sort of zigzags based on the exact geometry of how these chains work and the bond angles. So notice that if this thing here represents the overall tertiary structure, these coils and this little sheet section highlighted in blue represent specific structures within that overall structure. And so a secondary structure is sort of a mini three-dimensional structure that tends to show up in lots of different proteins. The final term is the quaternary structure. Not all proteins have quaternary structures, but occasionally, or actually fairly often, more than one chain of amino acids, so more than one polypeptide chain, can get all entangled and attracted together and bonded together to act as one protein altogether. And so when you have more than one polypeptide chain in a protein, we refer to that overall structure as the quaternary structure of a protein. So if this is one chain, you can see it now in red, entangled with all this other polypeptide chain, but they're actually two separate chains functioning together as one big protein. And that's what a quaternary structure is. The attractions for a quaternary structure are the same as the ones we listed for the tertiary structure. And just a little bit closer look at the secondary structures. So here's the alpha helix again, and you've got the different R groups that are able to form these hydrogen bonds with one another. And here's the beta pleated sheets with that zigzag pattern, and you've got the hydrogen bonding here between these shapes. So if you had to actually describe the difference between them, alpha helices form when very close together on the chain, you've got groups that can hydrogen bond, and so it coils up really tight. Whereas beta pleated sheets, those sections of the chain don't actually have to be right next to each other, as you can kind of see in this diagram. So the coils are the alpha helices, but the sheets, right? So it's this part of a chain, and then there might be a whole bunch of chain that's not involved in the sheet, and then you've got this part of the chain here, and so together these are forming the beta pleated sheet. And then it may go off for a while longer, and then you've got another section of the protein that is attracting and forming these hydrogen bonds with one another. So the parts of the chain that form a beta pleated sheet don't have to be like consecutive parts of the chain. They can come from different parts of the polypeptide chain, but the way that they attract together with the hydrogen bonds ends up creating that sort of flat pleated structure. So the two major categories of proteins, or some ways to think about them, is as fibrous proteins versus globular proteins. And so fibrous proteins are sort of, think of the word fiber. They tend to be quite long, and what's really dominant about them is their secondary structures, especially those alpha helices. They tend to be insoluble in water, and they tend to be structural components inside of biological specimens. So things like keratin, which makes up hair and wool and claws, but also collagen, which is important for connective tissues. Those are fibrous proteins, whereas globular proteins are more folded up and more spherical in nature, and their overall tertiary structure is more important. They tend to be fairly water soluble because the hydrophobic parts, any amino acids that have R groups that are nonpolar, will sort of naturally get folded in towards the center, while polar groups will tend to stick towards the outside. And so overall, the protein can dissolve in water. And these proteins do lots of different things, including they can act as enzymes. They can transport things through the blood because they can dissolve in water, and they can help send messages throughout the body as hormones. So if you look at the structure of hemoglobin, you see all these various coils and structures within it. And this is an example of a globular protein where it has this sort of spherical structure. And hemoglobin has a quaternary structure. It actually has 4 different polypeptide chains that all are interacting with one another as one giant protein. And so that's all we're going to get to in this video, which looks at primary, secondary, tertiary, and quaternary structures of proteins and the types of attractions that are responsible for them.